I'm Lisa Flanagan. I'm coming to you from the Department of Neurology and the Sue and Bill Gross Stem Cell Research Center, so I'm a little bit of a departure from the other folks that you've heard. I'm actually a biologist. And I want to thank the organizers for inviting me to come speak to you today about a project that I've been doing in collaboration with Abe Lee, who's part of the MF3 Center here, where we've been using microfluidics to try to improve the purity of stem cells. So I want to just tell you a little bit about why we care about that. So I think that stem cells in general hold a lot of promise for regenerative medicine. And part of the reason that that's true is because we can now go from these very undifferentiated pluripotent stem cells to basically any of the final differentiated cell types in your body. So the potential then of going in and replacing cells that are lost or damaged or injured from illness or disease is really pretty incredible. So what does that mean for us in terms of biologists about what do we have to do to make this happen? So there's a few problems that we need to solve, okay? So we know that if we want to transplant cells to cure some sort of a disease or replace cells that are lost to injury, we have to figure out what are the right cells to put in. So we know if we put in, for example, these very pluripotent cells, they'll turn into a tumor. We also know that for many systems, if we put in the very final differentiated cells, so for example neurons of the brain or spinal cord, they'll die. So what do we want to transplant? Well, the field has really been moving towards thinking about more specified progenitor cells or more specified even stem cells to put in for transplants. So if we think about how cells, for example, go from this very pluripotent state to the final differentiated cells of your blood, we can use that as a way to think about this. So people know now that it's really clear that there's a specific stem cell that forms in your blood system. It gives rise to a variety of specific progenitors that go on to form the final differentiated blood cells, for example, the red blood cells that you see over here. So my lab is interested in stem cells, but we don't study the blood stem cell system. We're interested in the cells that form your brain and spinal cord during development. And we are interested in seeing how these cells can lead to repair from things like stroke and Alzheimer's disease. So we know in this system that we go from a very undifferentiated neural stem cell to progenitor cells that have either decided that they're going to become neurons, which are the cells that form all the connections in your brain and spinal cord, or they're going to turn into, for example, an astrocyte, which is a type of glial cell. So this has been known from developmental biology studies for quite a number of years now, but we still have a lot of questions about what's the nature of these specific progenitors, what makes them different from each other, what's made this cell decide that it's going to go down this pathway. And part of why we have all these questions still is there's no good markers to really distinguish these cells from each other. They look identical. The kinds of markers that people have used in the blood cell system haven't really panned out very well in the neural system. So we were looking for a novel way to come at this problem. And that's when we got really lucky because we started talking to folks over here. We met Abe Lee, who's in the biomedical engineering department, and as I said, the MF3 Center. And he told us about something called dielectrophoresis. And I was very happy actually to see that word show up on one of Kumar's slides. It's nice to see it there. I'm not going to go through a lot of the details of exactly how this works, but I wanted to put this slide up just to say that what happens is we put a cell into a non-homogeneous electric field, and that causes a motion of that cell. I'll talk more about that on the next slide. But what was appealing to us about using this technique to apply to our stem cell problem is that it was label-free, so we don't have to have any particular markers that we know or identify our cells of interest. We can just go in a very unbiased way and just start probing our cells and see if there's anything useful there. So how can you use something like dielectrophoresis to separate cells or tell the differences between cells? Well, it's all based on the fact that there's a frequency-dependent response, okay? So basically, at lower frequencies, a cell will be in what's called negative DEP, which means there's a force that is repelling it away from these electrodes. As you get to higher frequencies, it's in positive DEP, so it's being pulled towards the electrodes. So if you have two cells that have inherent different characteristics, for example, our blue cell over here and our orange cell, you can pick a frequency at which one is in positive DEP, so it's being attracted to this electrode, and the other cell is in negative DEP, so it's being repelled away. So you can use that force then to drive the separation of the cell types. And there were a number of examples from the literature that people had used to use this force for separation, but just not really so much for stem cells. So we've, as I said, coupled with Abe Lee's lab, and he's introduced us also to using this in a microfluidic setting. And again, I won't go through the whole details of the experimental setup, but I did want to describe to you how we actually get these measurements. We basically take our stem cells, put them into a single cell suspension, and then they're flowing along in a microfluidic channel in a chip, and they're encountering these electrodes, okay? And at some frequencies, when we turn on the DEP force, none of the cells are in positive DEP, so they just all will flow by. At other frequencies, some of the cells will be attracted to the edges of the electrodes, the others will flow by. And at some frequencies, all of the cells in the mixture will be attracted along the electrodes. So when we first started doing this, we wanted to see if we could even see a difference between our very undifferentiated stem cells and the very fully differentiated cells of the central nervous system. And it turns out that we could. So you see here in black, these are the neural stem cells, and in blue, these are the differentiated neurons, and in green, the differentiated astrocytes. So that was promising that we could see a difference in those cell types. But even more interesting, we could see a difference now in our progenitor types. So I mentioned before the cells that will turn into neurons compared to the cells that will turn into astrocytes or glia. And using these DEP microfluidic devices, we can see a difference in those cells whether they've come from mouse or from human. We've gone on, and I won't go into a lot of details about this, but if you're interested, you can ask me about it later, to try to see how we could use the response of these cells to the electric fields to try to figure out what the differences are in the cells. And we've really been narrowing it down to changes that are happening in the outer plasma membrane of the cells. And it really looks like what's happening is the cells that are turning into neurons are starting to pick up some of the membrane characteristics of neurons, whereas the cells that are turning into astrocytes are picking up characteristics of astrocytes, which makes a lot of sense, but is helpful for us in terms of figuring out the key biological differences between those cells. We're now looking at the response of these cells in these electric fields as basically a label-free way of monitoring stem cell differentiation. So for example, we now know that we can start with a very undifferentiated human embryonic stem cell, see how that changes to now a much more differentiated human neural stem cell, and then as the cells start to bifurcate either on their way to becoming astrocytes or neurons, they also shift their properties. And so we're using this now as I said, a label-free way to monitor the differentiation of these cells. I mentioned before that people have used these types of devices and electric fields to separate cells, and we've been going down that path now as well. So what we do is we take a mixture of stem cells that we know have some progenitors that want to go down the neuron pathway, other progenitors that would like to go down the astrocyte pathway, and we put them through our fields. And what we're finding is a lot of the very undifferentiated cells get sorted out, and at low frequencies we're pulling out a lot of the astrocyte progenitors, and at higher frequencies we're pulling out the neuron progenitors. So this is, again, for us as biologists, this is really exciting because we now have a handle on these cells. We can start to learn more about their key characteristics because we can isolate them from each other. We started to compare this to the mainline sorting technology that most biologists use, which is FACS, which is fluorescence activated cell sorting. You have to have some sort of a label for the cells. Usually you use a cell surface label, often because you're looking at live cells. And we've been finding, at least so far in our hands, that our recovery efficiency and viability is actually much better for DEP than it is for FACS. Neural cells are very fragile, and neural stem cells are no exception. They don't actually deal that well with the fluid shear forces that are in FACS. So it's kind of ironic because we've been looking then at, okay, well, what are we talking about when you're running FACS compared to DEP? And if you want to run FACS, you have to have this really large, very expensive, complicated machine. And a lot of times, you know, we're lucky here at UCI, we have multiple of these, but very often you might find one per institution. But then when you look at our DEP setup, it's a little bit ugly, but you've got to admit that it takes a lot less space, it's much less expensive, it doesn't have all these lasers and things like that. We don't need to actually even buy antibodies, which is really nice. And so you can envision having one per lab, and I think that's part of what's appealing about this as well. So what have we learned then? So we now, you know, in terms of honing in on what are some of the key things about these cells, we're able to use these DEP microfluidic devices as a label-free and non-toxic way to isolate these cells so we can understand their biology better and use them for transplantation. And we're really excited to be part of the group here. Thank you.